So without further ado, I'll introduce the first speaker of the day, who is Dr. Aviv Mezer. So Dr. Mezer is an associate professor at the Edmond and Lily Safra Center for Brain Sciences at the Hebrew University of Jerusalem, Israel. After completing his undergraduate degree and graduate studies at Tel Aviv University, Professor Mezer moved to Stanford and worked with Dr. Brian Wandell as a postdoctoral scholar and research associate. And today he will give a talk titled Characterizing the Specificity of Quantitative MRI for Various Molecular Forms. So yeah, thank you very much, Dr. Mezer, for accepting our invitation and opening the conference. So yeah, and the floor is yours.
Thank you so much. Let me upload my presentation. Do we see the right screen? Can someone note? Okay.
Yes, all good.
Great. Okay, so thank you for the invitation. Thank you for the opportunity to present here. It's a great pleasure. It was a wonderful conference last year, and I'm looking on what is coming in the program. It seems to be a wonderful conference. So thank you. I'm going to talk about a molecular form that we can try to identify with quantitative MRI, and I will try to say how we try to validate that and how we try to assess our ability to actually measure those things that we try to measure. I would say also that I'm coming from the Hebrew University in Jerusalem, in Israel, and I'm sure you all know that we are actually in a war zone, in war right now, and we hope for it to end, and we hope for peace. We also hope for the coming back of the hostages in Gaza. So yes, with that, I will move to science. And I will quickly remind ourselves also, although we all know that quantitative MRI is really different than the normal MRI. And it's different in the sense that it doesn't look on the brain as a picture. So we don't using a MRI as a camera that's getting beautiful images of the brain, but as a technology that allow us to measure quantitative measure, physical measurement with physical units. And when we have that, it allows us also to develop method and models to assess the biophysics or the microstructure underlying the measurement. And it was termed in vivo histology, and it's a great name to what we try to achieve. And of course, when biology is complicated, of course, and when we have a complex system, we many times need to combine multiple measurements together, which it's by itself it's harder and also harder to then validate, and I will try to discuss that today. So that will be the topic of the talk. And when I say histology, with histology postmortem, we can do a lot, right? We can actually measure many aspects of the underlying biology, but only postmortem. I will discuss two today, so I will mention them first regarding histology. With histology, we can actually measure the lipid composition in the brain. That will give us an array of many lipids that exist in the brain and actually vary as function of age, for instance, and also in disease case. For instance, in this work, they were able to predict the age of the subject according to the lipid composition. So that's something we can do postmortemly. We can also look on iron, but not only on iron itself, but also the iron forms. Iron can be in different environment and interacting with different proteins. And this homeostasis essentially is actually critical, and it's changing in different stages, particularly in a neurodegenerative disease. So that's interesting. And we would like to achieve a similar level, or maybe better, or some kind of a similar level also with quantitative MRI, of course, in vivo. And that's hard, but that's the task. So I'm going to discuss today a work of a PhD student, Shir Filu, which developed this idea of relaxivity, which we find as a tool to be able to sense the molecular environment. And she did her work with two very important collaborators in the lab that did a lot of the experiments, Oshrat Stangel, which is a PhD student, and Rona Shabani, that's gonna present in this session later on a different and interesting topic about myelin organization. So first, I want to thank them when I'm describing this work. Okay. So here is a set of quantitative images. Each of them look different. They have different contrast. They have different units, and they all seem to be sensitive to the underlying tissue. They're all sensitive to the structural organization, the iron composition in the tissue, and myelin. And the combination is actually what it make it so hard. We don't know exactly how they are combined, but we know that all our measurements are sensitive to those forms or to this aspect of the biology, and all of them also sensitive to water, the fraction of water, because MRI is always a measurement of the water interacting with this environment. Okay, so I'm going to start with myelin. I'm going to do it very quickly, but I will try to explain the idea of relaxivity here, and then how we validate it. So we have here a set of quantitative measurement. We assume, and we have other earlier work that show that they're all sensitive to myelin in some way. So what does it mean? Let's take one of them, let's say the relaxivity here of R1 fixation. So let's say we have two measurements, and one has a higher value than the other. What we assume when we say it's sensitive to myelin is that if there is more myelin, so if there is an XX of concentration, then when there is more myelin, the R1 is greater. So that's the idea here. But the thing I want to consider here and add is that in some case, we can imagine a situation in which the concentration is the same, but the relaxation change. So in this case, we have some kind of uncertainty, essentially. But it also can be a point of value if we would know to tease apart the different sources. So that's the point here. And this is so it can allow us to increase the specificity if we would know to tease apart the different source. And I would like to argue that lipid composition could be one of those important sources. And for that, I need the idea of relaxivity. Relaxivity is the ability of certain compounds, say a lipid, to increase the relaxation of the surrounding water protons. So assuming we'd have multiple points here in different concentration of this material and this material, then what we would see that they are following a different line. They're following a different line, and this line have a different slope, and this slope is the relaxivity. So if I would like to put it in a very simple equation. So say we're measuring R1, and it's sensitive to this concentration, and it's sensitive to concentration by multiplying with this parameter, which will be the relaxivity. Okay, so the slope of this line is the relaxivity, and this is great. This is a signature of the material of the molecular form, but here's a problem. We actually need to know the concentration in order to find it. So that's something we can only do ex vivo or in vitro. And the question, can we also do it in vivo? And my argument is yes, and I will show you how and how we did it. We're actually using another quantitative measurement. So using more than one quantitative measurement here. And here we're using the tissue fraction, which is a measurement that's coming from water content. It's a measurement that quantitative MRI measurement coming from proton density and then normalized. We do 1 minus of that. We ended up with this lipid and macromolecular tissue volume, which is the fraction of non-water, essentially. And this is a quantitative measurement and can be validated. For instance, here, it's a validation that's done in histology. Each point here is a different position in the brain of a post-mortem fresh brain scanned. And then there was a histology measurement in which we're just measuring the fraction of water. And you can see that it's a very good measurement of the fraction of water in the brain, fraction of non-water. Now, when we have this measurement, we can actually replace here the concentration with MTV. And now we have an equation of R1 and MTV. Both of them we know to measure in vivo. And because we know to measure them in vivo, we can actually extract this relaxivity. So we have the tissue relaxivity, which we suggest is sensitive to the molecular form. So now, Shir in her article went through this cycle, and I will do it very quickly, just emphasize the validation aspect in it. So we have here a biophysical model, right? We just saw a biophysical model that's supposed to be sensitive to the molecular form with MRI. So the first thing to do for us was to go and actually manufacture this environment in vitro, creating phantoms of different phospholipids and see what would happen. So that's exactly what Oshrat Stangl did. She measured different phospholipids. Here we see two, and those two are very common in myelin. And each dot here is a different concentration. She changed the concentration, so MTB change, R1 change, and we can see that different phospholipid has a different linear relationship. So essentially goes with the theory that this relaxivity or this slope is actually sensitive to the molecular form. Next, we move to in vivo, measure the same thing in vivo across different brain regions. And here we compare the relaxivity across brain regions, that's what we see here on the x-axis, to lipid composition as we found in the literature. So we did a comparison between lipidomic analysis that was done in the literature to what we measure in vivo. So this is different brains, but at least the difference across brain regions seem to be similar to what we measure in vivo in the human brain. Now this is good, it's a group analysis, but we also wanted to have a single subject analysis and do the same thing on the same brain. And for that, we actually measure porcine brain. There were fresh postmortem brain in which we scan in the MRI, took biopsies. So each point here is a different position in the brain, send those positions to lipidomics analysis. We got this array of lipid composition and compare the lipid composition to the MRI measurement, to the relaxivity. And here again, we find now on the same samples that the relaxivity goes with the lipid composition. And that was our way to try to validate and enhance our belief that this relaxivity is actually sensitive to the lipid composition. So that was a work that was published already a few years ago. And now I want to describe a work that was published just really recently, like just a few weeks ago, which would have a very similar taste, but on a different aspect. And this is the work that relate to iron. So also in the case of iron, we know there are several measurements that are sensitive to iron, particularly to the amount of iron content. So now iron is very important material in our brain. It's actually, the homeostasis of iron is critical, and it's really strictly saved and important for our brain physiology. And there are two main players in this, is ferritin, which aggregate the iron within the cells. And there is transferrin that transfer it between the blood and the cells. And there are more, but they are the two major players. And any change in the iron homeostasis goes with some problem in the brain, and it could be a signature for different diseases, such as neurodegenerative diseases and others. Okay, so we really would like to measure this iron homeostasis. That would be nice. Now, I said before, we know to measure, or we know to estimate the iron concentration in the brain with MRI. This is all news. Many did it. Here we did it again. Here we compare iron concentration. This is a measurement that comes from histology of other groups across different brain regions. Different points here are different brain regions. There is a point that comes from younger subject and older subject. And this is autostar measured in vivo. So this is a replication of work of others that show that measurement like autostar, we can also probably do it with QSM, would show a good relation between the relaxation and the concentration of iron. What we wanted to get, and we don't have yet, at least we didn't have when we did our work, was a way to measure the molecular form of the iron. And I'm going to talk about our version, but I also want to highlight the work of Evgenia that's going to talk in this conference, I think on Friday, if I remember correctly. And she's also trying to tease apart these different sources, so it's going to be very interesting to hear her as well and read her articles. Okay. So our attempt was as follow. We said there are several quantitative measurements that are sensitive to iron, but they are sensitive in different way, and they're very different mechanism. In particular, we look on R1 and R2 star. They're both sensitive to iron, but in a different way. While R2 star is really sensitive to the mesoscopic effect of iron on the tissue, R1 is really sensitive to the molecular interaction of iron with water. So it's a very different mechanism. So we see also very different relaxivity here. And we thought that we can use it in order to assess the iron form. So I'm going to use the same cycle here again. First, let's look on the biophysical model. It's the same. We have here relaxation rate. It could be either R1 or R2 star. And now in X we have a iron concentration. And we have different forms. We assume there will be a different relaxivity. And this is something we can only measure ex vivo or in vitro. Let's put it in the equation again. Same equation of R1 or R2 star. Each of them is dependent on iron concentration with a relaxivity parameter that we don't know, and we can estimate only ex vivo or in vitro. Now, what she did, she said, these two measurements we have in vivo, can we rearrange the equation in a way that will allow us to measure it in vivo? And we can. So what she did here, she put R1 again, R2 star now. So those are two measurements that we can measure in vivo. And the prediction of the model is that we're also going to get a linear relationship here between the two measurements. And if the equations are correct, then R1 will be a linear relationship of R2 star, and the coefficient will be the ratio of the relaxivity of each of them. This is useful, and we want to test that. So let's see how we test it. So first we went to in vitro. Here is the work of Rona that did the experiment. She created different environment of iron, either ferritin, transferrin, with lipids, without lipid, different environments. Each color here represents different environment. Different dots are different concentrations. And we can see here R2 star, we increase the concentration, R2 star grows, also R1 grows. And you can see that the slope here, the relaxivity here, this iron relaxivity, change as for different iron environment. So this is good. This goes with the theory. You can actually measure the R1 to R2 star relaxivity, and we're going to see that different environments have a very different relaxivity. And we also have a prediction from the theory, so we actually can check the theory here. If those values is the ratio between the two relaxivities that we measure in vitro, and the answer is yes. In many cases it is. There is a very good agreement between the theory and the measurement. Great. Now in vivo, we can measure also the relaxivity in vivo in different brain regions. Here I'm showing here 4 regions: white matter, thalamus, putamen, and cortex. You can see here R2 star, again R1 against R1. And again, each of them have its own linear dependency, essentially different iron relaxivity. So we can ask, is it goes with what we know about iron forms? So this is the validation against ex vivo and comparison to ex vivo data. So we saw this figure before. R2 star, we saw before is really sensitive to the iron concentration, right? What would happen with the R1 to R2 star relaxivity? So it actually is not very sensitive to the iron concentration, but it's sensitive to something else, which name here iron mobilization. What is iron mobilization? It's a measure that is measured not by us, by others. So this is data that we aggregate from other articles, which is the rate, essentially is a measurement of the ferritin to transferrin ratio in the tissue. So we find here that the same variation that we find here of iron mobilization coming from a post-mortem data goes with what we find here in vivo across brain regions. Here we also want to do, again, a measurement within the same subject and not just a group level analysis like that. And here we built this environment, which I think is interesting and pretty unique. And I think it actually could be used by others as well. And for doing that, we actually collaborate with a neurosurgeon, Tal Shachar. And what we did here, we scan patients which have brain cancer. You can see here the meningioma. So those subjects anyway are going to get scans, and we add quantitative scan to their scans. Now these tumors are going to be resected. So as part of the clinical procedure, the tumor are resected. And then what we did is so we have a quantitative measurement, the tumor is resected, so we kept it essentially and send it for a histological analysis. Particularly in this case, we measure the transferrin to ferritin ratio within the tumor of each subject. And so we could compare it to the measurement that we have. What did we find? We find that if you look on this iron mobility, which is the transferrin to ferritin ratio, we had two groups in our cohort of a cancer patient. Some of them have high iron mobility, the other have lower iron mobility. And if we checked which measurement actually can capture it, it was R1 to R2 relaxivity. It actually separated between the two groups, the one that have high iron mobility and low iron mobility. That was significant, and it wasn't if we would use R1 or R2 star. So the core measurement that created this relaxivity couldn't do this separation. So essentially, we add here more information, and this is how we validate it. Last thing I want to share with you, because I think it's interesting, because we already have this data, we have these tumors. We actually collaborate with another colleagues, Nomi Habib and Masha Adam, which sit here in the lab next to us, and they are pro in RNA sequencing. So they took this sample and sent them, actually did RNA sequencing on those samples. So we have 15 tumors that have been sent to RNA sequencing, and then we can actually look on the different families or annotation of families of genes that are enhanced in relationship to the values of our quantitative measurements. So we have here three quantitative measurements, right? R1, R2 star, and the relaxivity. And when we do this clustering analysis, we can actually see that the measurements are very separate. There's a great separation between them in terms of clustering. So that gives us a sense that this new measurement is really new because it actually capturing something different in the biology. And the interesting thing is that not only that it was different, that there was many iron related family of genes that were in this R1 to R2 star relaxivity and that pops only in it and not in the others. So that's another form of validation. In particular to show that it's a new measurement which sense a different aspect of the biology. Yes. So I will summarize actually. In the last part, I showed you how we first develop a biophysical model, and then we use a experiment, in vitro experiment, bottom-up way of analysis to actually validate the prediction of the model. And by that, we validate that R1 to R2 star relaxivity was sensitive to the iron forms. We did then a comparison between the measurement in vivo to non-measurement ex vivo across brain regions. We then took the effort and measure it within the same subject. That was the work that we did on brain cancer, in which we scan before surgery and measure after surgery, the biology. And actually, this cycle, it was very similar to the cycle that I showed you before of validation for the lipid composition. So it's the same kind of a way of thinking. And with that, we hope we present a way to first enhance the specificity of MRI and also to validate it in different ways. And with that, I would say thank you again. The work is, as I said, the work of Shir Filu, her PhD, with a lot of help of Oshrat and Rona. Our collaborator Tal Shachar and Nomi, funding agency, of course, and thank you for listening. That's it. Took me 23 minutes.
All right. Thank you very much, Dr. Mezer. That was a fascinating talk. That was really, really interesting. Chris, do we have any questions in the chat?
Yes, we have one from Jerry Zhang. So I wanted to know, it seems that a few quantitative measurements are sensitive to both iron and lipids, as you sort of introduced. So if we want to estimate a specific molecular content, such as iron, do we have to exclude the other molecular content's impact on the quantitative measurements? And if so, how would you do that?
Right. But essentially, the question is even harder than what I try to answer, right? Because I try to kind of separate the sources by looking on the dependency, essentially saying what I did here. I say, for instance, in the case of the lipid, I say, R1 is sensitive to many things, I will ask what is the dependency on something which is only the fraction of water, which I assume have to do with lipid and other macromolecules actually. But if we actually want to take the measurement and tease apart the different sources, then we actually have to build the linear model, and I think it's hard. And so essentially build the different sources into one equation to fully explain R1, R2 star, etc. I think our only, and I didn't show it, but in some experiment that Rona did, actually, those things actually also interact non-linearly. So it even make it harder. I don't think we have a good, easy solution for this question. Besides, some measurements are more sensitive to one measurement than the other, one source of the biology than the other. And so that's what we typically do, right? We notice that R2 star or QSM are very sensitive to iron and also sensitive to other, but less, etc.
Thank you. Any other questions, Chris?
None from the chat, but I'll ask one as well. So we've had some discussions in terms of, so you have R1, R2 star, or you could take the inverse T1 or T2 star. And that sort of comes back to how you're modeling this with the linear relationship. So you mentioned that sometimes it looks like the linear relationship doesn't hold. And given that these parameters are fit to sort of exponential signal changes, do you expect the linear relationship to hold as well if you were to look at the inverse instead? So the T1 or T2 star.
Right. So it's not supposed to, but sometimes it does, by the way. And it has to do with the, first on the range. A thing which are in short range, any exponential in a short range can be approximate with a linear fit, right? So then the 1 over would be still the same. So I think it's really depend on the range. But things are, according to theory, things are summed linearly in R1. And so by definition, they wouldn't sum linearly if you use the time constant. But unfortunately, given the noise and the variability of our that we actually experience in the biology or in the brain, sometimes it actually doesn't make a huge difference if you use T1 or R1. But so the theory versus reality, but yeah.
Thank you. We actually have one more question here from Yasik. With multiple methods sensitive to myelin content, what is your opinion on joint inference and which multiple methods are acquired, such as MPM, diffusion-weighted imaging, or MTR, and they're all collectively entered into a PCA analysis to extract one big myelin component?
I'm not opposing, right? I'm also doing that sometime. So we have all this measurement and we just let the data tell us essentially by PCA or other forms. And sometime it will work. And I'm finding, well, I'm fascinated by this biophysical model and try to tease apart sources. And I think, well, so at first, all this PCA or other kind of big data analysis and then sorting different sources sound to me like a good approximation, and many times it will work very well. And I find it more, not more, but I'm fascinated by the approach of trying to find the different aspect of the biophysics and try to model them. And I actually think that they can also be input for such a model. So if we have a smart way to combine the different parameter, I think it can help us also in building this bigger model. So that's the way we look at it in my lab.
Great.
All right,
Thank you.
thank you. Yeah, that was really interesting, really interesting. So I think now, unfortunately, we have to move on to the next talk. Thank you again, Dr. Mezer.
Thank you.